السلام عليكم ورحمة الله وبركاته. بسم الله الرحمن الرحيم. This is the third lecture in cytology. Today we are going to talk about lysosome, which is a membranous organelle responsible for digestion. The lysosome appears as spherical membranous organelle. It is the digestive apparatus of the cell. Here, this diagram shows the relation between the rough endoplasmic reticulum and the Golgi apparatus in releasing the lysosome. The hydrolytic enzymes are synthesized inside the rough endoplasmic reticulum and released as transfer vesicle. This transfer vesicle enters the Golgi apparatus from the cis phase, releasing from the trans or the mature phase the lysosome. So again, the enzymes originate from the rough endoplasmic reticulum, and the lysosome membrane originate from the Golgi apparatus. The enzymes that present inside the lysosomes are phospholipase, protease, acid phosphatase, nuclease, and sulfatase. Dear students, the word ase digests what comes before. For example, sulfatase, it digests the sulfate, and so on. The number of lysosomes increase in phagocytic cells. Okay. This is a phagocytic cells. From where I know that it is a phagocytic cell, the cell membrane is irregular as it forms what's called the pseudopodia in order to digest a foreign particle. When it digests the foreign particle enter inside the cell, the lysosome start to react with the digested substance. The phagocytic cells as white blood cells, neutrophils, and macrophages. By LM, the lysosome cannot be demonstrated. It should be demonstrated by histochemical method, detecting the enzyme present inside the lysosome, which is, for example, acid phosphatase enzyme. This method is called histochemical method. I cannot see the lysosome by the light microscope. I can detect the enzyme which is present inside it. By the electron microscope, I have two major types of lysosome. Primary lysosome, which is the newly formed, which released from the mature phase of the Golgi apparatus, that have not entered any digestive events. It appears spherical, surrounded by a single membrane, and it appears homogeneous vesicle. This is an EM picture of the primary lysosome. Here, as you see, it is rounded, membranous organelle surrounded by one membrane, and the content are homogeneous. Here is an EM picture of lysosome. I can see different types of lysosome. This is homogeneous, this heterogeneous, and this, and this again. All these are heterogeneous, which are the secondary lysosomes. Here again, this photo, please see here. As we said, the transfer vesicle released from the rough endoplasmic entered the Golgi apparatus, releasing lysosome. This lysosome is called primary lysosome. If the primary lysosome react with a phagosome that enter by phagocytosis, pinocytic vesicle which enter through the pinocytosis, or if it reacts with an old organelle, it will change into secondary lysosome. So secondary lysosome is primary lysosome involved in digestive activity. For example, the heterolysosome is fusion of primary lysosome with phagosome. Multivesicular body is fusion of primary lysosome with pinocytic vesicle. Autolysosome is fusion of primary lysosome with autophagic vacuole containing old organelle. This old organelle, I want to get rid of it because it's dangerous to the cell to have something like this inside it. So we should get rid of it. Again, EM picture of secondary lysosome. Here it is heterolysosome, which is fusion of the primary lysosome with phagosome. Here, multivesicular body. I can see something like fluids inside multiple cavities. Multivesicular body. Again, This is heterolysosome, and this is primary lysosome. Again, heterolysosome. What is the autolysosome? Fusion of primary lysosome with destroyed endogenous substrate, for example, old mitochondria. For example, old mitochondria. After digestion, there is a residual body. This residual body contains indigestible compounds. What is the fate of this residual body? Either released by exocytosis, and this is called cytosol, or accumulation in the form of lipofuscin granules, which are called age pigments. These age pigments present in old age cell, for example, the nerve and the heart cell. You know that the nerve cell and the heart cell does not multiply, and the number is constant. Okay, other cells short life as the phagocytic cells. They can live only 3 to 4 days. So this undigestible substances present in long age cell as the nerve and the heart cell. Okay, this is the here it is autolysosome, phagolysosome, multivesicular residual body. Diagrammatic picture. Okay, again, this is the rough endoplasmic reticulum, Golgi apparatus, releasing primary lysosome. If a primary lysosome reacts with autophage, called autolysosome. If it reacts with panocytic vesicle, it will be called multivesicular body. If it fuses with phagosome, it's called heterolysosome. The end result is a residual body. This residual body either we get rid of it by exocytosis or remain inside the cell in the form of lipofuscin pigments. Fate of the residual body, discharge of the content by exocytosis, or stay in long-lived cell, for example, the cardiac cells, in the form of lipofuscin pigments. What is the function of lysosome? The main function of lysosome is digestion. Digestion of what? Nutrition. Bacteria. I can phagocytose bacteria, so it has a defensive function. Fluid. Old organelle, degenerated organelle, to maintain the cell healthy. Digestion of all our body, what's called autolysis after death, when the body when death occurs, the pH of the patient decrease, and the lysosome rupture, releasing digestive enzymes, causing digestion of whole body. Help the sperm to penetrate the ovum. You know that the ova is surrounded by a thick wall. This thick wall should be penetrated by many sperms. The sperm head carries hydrolytic enzymes that dissolve this wall that surrounds the ovum, helping its fertilization. In the thyroid gland releases hormone called thyroxine, T3 and T4. They are secreted in a large amount, so they are stored inside the thyroid gland. In need, I need the hormone, storage of the hormone, a combination of the hormone with a glycoprotein. This combination should be split by the action of lysosome, breaking inactive form to active one. This inactive, the stored form, into the active one, which is the free form. Medical syndromes develop due to lack of lysosomal enzyme, lack of sulfatase. I said before, if I have no sulfatase, I will not break the sulfate, results in intracellular accumulation of sulfated compounds which interfere with the normal function of the nerve cell. Fusion of autophage vacuole with lysosome is called autophagolysosome. other organelle, membranous organelle, called the peroxisomes or the microbodies. By definition, it is a self-replicating organelle as the mitochondria, contain oxyreductase enzyme, which is the amino acid oxidase and catalase. Can be demonstrated by histochemical methods as that of the lysosome. By EM, they are spherical ovoid membrane-bound organelle. The precursor vesicle, the vesicle buds off from RER, while the enzymes are synthesized by free polysomes. Then the enzymes will enter the vesicle by something called internalization, as the membrane that released from the RER containing receptors that can identify the enzymes. Again, the free polysomes produce the enzymes. The vesicle of the peroxisome is produced by budding of the RER. This vesicle has receptors that will know the enzymes entering to it. This is called internalization. The peroxisomes, what is the function of the peroxisome? Beta oxidation of long-chain fatty acid by the action of oxidase. This reaction will produce energy. This energy comes in the form of a heat and not stored as ATP, not as the mitochondria. After production of this oxidation, hydrogen peroxide will be produced. This hydrogen peroxide is toxic to the cell. We get rid of the hydrogen peroxide by the action of the catalase, which presents in the peroxisome, breaking the hydrogen peroxide into water and oxygen, changing the hydrogen peroxide, which is toxic, to non-toxic substance. H2O2 by catalase change into water and oxygen. The oxidation are important in the liver cell because it causes detoxification of alcohol and some drugs. Deficiency of the peroxisomal enzymes affect the structure and function of some organs as liver and kidney. How to know that there is deficiency? By the electron microscope, I can see empty peroxisomes. Now we are going to talk about the non-membranous organelles. What the meaning of non-membranous organelles? They are permanent, essential, vital, and have a specific function, but not surrounded by a membrane. For example, the ribosome. By definition, it is a small non-membranous body measuring 20 multiplied 30 nanometer, formed of rRNA and protein. Okay, this rRNA is formed inside the nucleolus, and the protein is formed in the cytoplasm. The protein moves from the cytoplasm to the nucleus through the nuclear pore. In the nucleolus, the small and the large subunits of the ribosomes are formed, passing to the cytoplasm and join during protein synthesis. Again, the protein synthesized in the cytoplasm move from the cytoplasm to the nucleolus, combine with the rRNA, forming the small and large subunits of the ribosome. The small and the large subunit of the ribosome passes to the cytoplasm and fuse only during protein synthesis. The number of ribosomes abundant in protein synthesizing cell. Okay, by light microscope, it causes basophilia. Why? Due to presence of the phosphate group, which is negative charge, stained by positively charged stain, which is the hematoxylin. This basophilia may be diffuse, as in precursor of blood cell, may be localized here at the base of the cells of the pancreatic acini, may be focal, forming what's called Nissl granules in the nerve cell. So the ribosomes causes cytoplasmic basophilia because the ribosomes are RNA, ribonucleic acid, and the acid like to be stained with something basic. and something basic, it's called basophilic, and the basic stain is blue and called hematoxylin. The ribosome by the EM is formed of two subunits, small subunit and large subunit. The large subunit is divided by a groove. The small and the large subunit only unite together during protein synthesis. When the ribosomes attach it to the messenger RNA. This is the small subunit, here the large subunit. The large subunit is divided into two halves. Okay, this is one ribosome, one and other one, joined together by messenger RNA. The ribosomes may be free, singly, or polysomes connected by the messenger RNA forming rows or spiral, or may be attached to the rough endoplasmic reticulum. Here are ribosomes, small subunit, large subunit, attached together by the messenger RNA, producing protein. This protein directly goes to the cytoplasm. Attached ribosomes, the large subunit of the ribosome attached to the rough endoplasmic reticulum by a receptor known as ribofaurin. They are also attached to messenger RNA that carry the information how to form the protein for the amino acid sequence to synthesize the polypeptide chain. Transfer RNA will pick up the specific amino acid and transport them to the ribosome. Each ribosome read and translate the code, forming the protein that extends down through the groove to the lumen of the rough endoplasmic reticulum. Again, here is the small and the large subunit of the ribosomes. They are attached by the messenger RNA. This messenger RNA carries the amino acid sequence from the DNA that's present inside the nucleus. The small and the large subunit fuse together only during protein synthesis. The large subunit attached to the rough endoplasmic reticulum by the ribofaurin. Then the transfer RNA will pick up amino acids, giving them to the ribosome, forming the polypeptide chain that will enter inside the rough endoplasmic reticulum. What is the function of ribosomes? Protein synthesis. If it is free or polysomes, they are needed for the growth and regeneration of the organelles and forming enzymes. If it attached, they form a protein that will be secreted out of the cell, protein for export, as hormones, albumins, and others. Here is the attached ribosomes. Ribosomes joined together by the messenger RNA attached to the rough endoplasmic reticulum through the large subunit by attached to the rough endoplasmic reticulum by the ribofaurin, and this is the protein that will be produced inside the rough endoplasmic reticulum.